Welcome to this video on the Demystifying Research channel, where today we are going to have one of our very own McMaster experts on to talk about the production of influenza A vaccine. So welcome, Dr. Mullarkey. Thank you for joining me today.
Thanks for having me, Julia.
So let's start off by having you just tell us a little bit about yourself.
Sure, absolutely. So my name's Caitlin Mullarkey. I'm a faculty member in biochemistry and biomedical sciences. And my background is in influenza viruses with a particular emphasis on vaccine development and how to make better influenza virus vaccines. As we'll discuss today, influenza vaccines are really unique among vaccines in how we have to produce them and reformulate them every year. So I completed my doctoral work at the Jenner Institute at the University of Oxford, where I worked on a viral vectored influenza vaccine strategy. And then I moved on to do my postdoctoral fellowship with Dr. Peter Palese at Mount Sinai in Manhattan. And my work there also focused on a novel influenza vaccine strategy. And there I was really interested also in understanding mechanisms of a particular class of antibodies and how they can protect against flu, how they can offer better protection against flu than the antibodies that are typically elicited by seasonal influenza vaccines.
Our first question is, why is influenza important to study and make vaccines against? Why is it so important to be able to prevent infection?
Sure. And I would say pre-COVID, this was probably something I didn't have to convince people of. But since 2020, when the respiratory virus that has absolutely dominated both our global attention and our global public health is SARS-CoV-2. Influenza virus is still a global health problem. And despite studying influenza viruses for hundreds of years, we've known about influenza for hundreds of years, they still pose a problem every year during the winter months. So maybe I'll take a step back and say pre-COVID, globally, there were around 500,000 deaths as a result of influenza, right? Which maybe does not sound like that much, again, in comparison to what we see with SARS-CoV-2. But that doesn't tell the whole story of the burden of influenza, because there's lots, even though there were hundreds of thousands of deaths, there are millions and tens of millions of cases. And this can really put a burden on the healthcare system, on the hospitals, and also from in thinking about workplaces, there's a big problem with absenteeism, right? People get sick and they can't go to work. In the era of COVID, we've actually seen until recently, I'll put this as a caveat, decreased influenza virus activity as SARS-CoV-2 has dominated. Some of the reasons that explain that are behavioral, right? Masking policies, social distancing measures have decreased flu transmission in the past two influenza, kind of what we think of as cold and flu season. But there is currently, it's November now, we're heading into cold and flu season, and there is increased influenza virus activity at the moment. And this is the first year I think, and other experts think, that we're actually going to see a lot of co-transmission of SARS-CoV-2 in influenza viruses. So it causes seasonal epidemics, but I'll also point out that influenza viruses occasionally cause pandemics, right? And this is when we see a new virus emerge that hadn't previously been seen before, people don't have any immunity to, and then we have, again, similar to SARS-CoV-2, a big global health problem. In the 20th century, we saw 3 influenza virus pandemics. So in the latest one being in 2009. 2009 was the last influenza virus pandemic.
We know that vaccines are one of, if not the most effective mechanism to prevent infection of different pathogens such as influenza. So knowing that, what are the major components of a vaccine? Let's just go over what kind of makes up a vaccine.
Yeah, so lots of different flavors for vaccines. Vaccines can take a lot of different forms. Some of the oldest vaccines are live attenuated pathogens. So you take the actual pathogen that you're trying to create a vaccine against and you weaken it. And then there's lots of other different flavors really that are just pieces of the vaccine. In some cases, you can have sort of what we call virus-like particles or empty shells. So, lots of different forms of vaccine can take, and more recently we're seeing things like mRNA vaccines, and certainly people have been studying DNA vaccines for a long time. Those aren't licensed. But the long and the short of a vaccine is that it shows your immune system that pathogen. It can either be just a piece of the pathogen. Sometimes it's the entire pathogen itself, depending on the particular vaccine. And then by showing your immune system what this pathogen looks like, you can develop an immune response such that when you were to be exposed to that pathogen after you receive the vaccine, you would have a very robust immune response, and that would help protect you from severe disease, death.
And so in terms of influenza in particular, what type of vaccine is an influenza vaccine that you might just receive at the pharmacy typically?
Right. Yeah. So most seasonal influenza vaccines are what we call detergent purified vaccines that are produced in eggs, right? And there's a really interesting process through which the influenza viruses are grown in eggs and then purified to enrich the vaccine for certain components. So when you go to the pharmacy to get your seasonal flu vaccine, most of the vaccine nowadays are quadrivalent, which means they have 4 different types of influenza antigens in them from 4 different influenza virus strains. For influenza, the principal component of the vaccine is the surface glycoprotein of the virus. So influenza virus is an envelope virus. It has a negative single-stranded RNA genome. And one of the 2 main antigens on the surface is something called hemagglutinin. And hemagglutinin is actually how the virus gets into our cells, right? It's the glycoprotein that is used for entry. So our influenza viruses contain 4 different types of HA. Typically, it is 2 different influenza B components, and then there is an influenza A H1 component and an influenza A H3 component, right? So those are the strains that have sustained transmission in humans.
And you mentioned the production of this viral antigen component of the vaccine. So thinking about how it's produced in embryonic chicken eggs, I imagine that would be an incredibly large operation considering the number of influenza vaccines we need out.
Yes, it's a very laborious process. It's very long, right? And this is why influenza vaccines are unique. And so maybe I can just take a step back and say that influenza vaccines are unique in that every year we have to reformulate them. And the reason for that is influenza changes all the time. It's very, very good at making very small changes to the surface glycoproteins such that it evades our immune response, right? So each year we have to predict the strains that are going to be circulating in the coming flu season and then make a vaccine that best matches what we think the circulating strains would be. Sometimes we get this right, sometimes we get this wrong. And the reason that we have to pick the strains typically 6 months in advance is that's how long it takes to produce the vaccine, right? So the strains that are in our current vaccines that are being rolled out in the clinic now, September, October, November, all the way through flu season, those were picked back in February, right? Because that's the lead time we need to be able to produce the vaccines. So again, each year we're reformulating and making these vaccines new. And that's different than any other vaccine that we have. We don't do that for any other vaccine. So it's really what can be challenging about having good vaccine efficacy is the fact that we need this big lead time to produce the vaccine. Influenza changes a lot. I can tell you about how we pick the strains, but there are limitations to how the data that goes into that. And so sometimes we get it wrong and there's a mismatch between the strains that are circulating and the strains contained in the vaccine. And then our vaccines have lower efficacy. even when they're very well matched. So that means when the strains in the vaccine are well matched to the strains that are circulating in flu season, the vaccine efficacy is around 60% in healthy adults. So there's certainly room for improvement, but this is what we've been doing actually for decades now. The way that we make influenza virus vaccines has not changed in a very long time.
So in terms of actually making the vaccines, producing, this massive amount of doses, could you walk us through the process of cultivating the virus in embryonic chicken eggs?
Yeah, sure. And so what I'll do is I'll talk about how we typically do this in a research setting, and then you can think about how that might scale to vaccine production to produce hundreds of millions of doses. So just to give you a frame of reference, the US typically makes 150 million doses of influenza virus vaccine, and one egg can produce one dose of vaccine. So if everything went perfectly and there weren't any hiccups and the yield was 100% all the way through, that means you need 150 million eggs to produce the number of vaccines that are needed during cold and flu season. So there are constraints in relying on an egg supply to make a vaccine. There are also issues related to adaptations of the virus in eggs that perhaps make them less immunogenic in humans. And then kind of further on to that, growing the vaccine in eggs can also be problematic for individuals that have severe egg allergies. And I will say that not all influenza vaccines are grown in eggs. There are some cell-based vaccines. The majority are, though. The majority of what people will receive as a seasonal influenza vaccine would be produced in eggs. So influenza virus is actually a pathogen of waterfowl. The natural reservoir for influenza viruses are waterfowl. So maybe that helps explain why we grow them in embryonated chicken eggs, is because this is the natural reservoir for the virus. The virus likes that environment, and it will grow to very high titers, which is just another way of saying amount. It grows to a high amount in these embryonated chicken eggs. So in order to grow the pathogen in chicken eggs, you actually put a small amount of virus in the fluid that surrounds the embryo, right? So again, these are embryonated chicken eggs. That means they're fertilized. So they're different than the eggs that you would buy in the grocery store. So typically, if you have an embryonated chicken egg, you'd have a small amount of the virus that you want to inoculate in those eggs. And remember, each year, the vaccine typically contains 4 different strains of influenza virus. So you have to inoculate eggs with these 4 different strains. So you have a small amount of the virus. You typically would visualize the embryonated chicken egg with some sort of light source. So you try to illuminate it, and that helps you see where the embryo is and where the fluid surrounding the embryo is, because your goal is to get the influenza virus in the fluid that surrounds the embryo. And typically you would mark the egg, you would visualize the embryo, the fluid, and the air space in the egg, and you might make some markings on the egg to, once you remove the light source, to remind you where those particular landmarks were. Then you have to actually breach the shell, right? So you have to make a small hole in the shell, which is a delicate operation because you can easily crack and make a very large hole or completely compromise the embryonated chicken egg. So that can be done with different tools. Different labs might use different tools, and in a industrial setting where you're inoculating millions of eggs, sometimes this is done with robotics and specialized equipment. So you would breach the egg, you would inoculate it with a small amount of virus, and then typically you're going to seal the hole through which you've just made in order to keep the environment as sterile as possible. In the lab setting, sometimes we just do this with a little bit of wax. So we heat up some wax, put some hot wax on the hole, and then when it hardens, it covers the entry point very nicely. Sometimes some people honestly just use a little bit of glue over the hole. So that's how you actually get the virus into the embryonated chicken egg. Then typically you would incubate that embryonated egg for about 48 hours at 37 degrees. And after which point in time, you would move that embryonated egg into a colder environment. So you'd go from 37 degrees, which is quite warm, to 4 degrees. And moving it from 37 to 4, the purpose of that is to actually kill the embryo, right? So that when you go to harvest the fluid, the embryo is no longer living. So after you have moved the eggs to 4 degrees, typically you'd leave them there for 24 hours, some amount of time. Again, you need to breach the shell, and now you need to take out all of the fluid that surrounds the embryo, which can be a little bit tricky because there's a lot of membranes and other things that can also come up with the fluid as well. And so you would remove the allantoic fluid. And remember, at that point, that is in that preparation, that is live virus, right? It's the whole virus. It's very much alive. It hasn't been undergone any sort of procedures to kill the virus, or to split it up into its components, right? Influenza viruses can grow to very high titers in eggs, something like 10 to the 8, 10 to the 9 virus particles per mil, and you can get in the best case scenario, you might be able to get 10 mils out of an egg. So really, really large amounts of virus come out of that egg. Then in terms of vaccine preparation, we don't use live virus for influenza vaccines. We kill the virus and we purify it to enrich it for that hemagglutinin component. And that's typically done. There's lots of different ways that that's achieved, but the purification is usually a detergent purification that enriches for the HA component. What I'll point out, though, is although we license influenza vaccines on the amount of HA that they contain, and for reference, that they have 15 micrograms of each strain. So in total, they have 60 micrograms of HA, right? Because they're going to have 15 from an H1, 15 from an H3, and then there's typically 2 B viruses in there as well. Even though we enrich them for HA, there are some other viral components that make it into the vaccine as well. Some other proteins like neuraminidase, which is another surface protein. So that I don't want anyone to think that the only thing in influenza vaccines is the HA component. There are other viral proteins that make it in. It's just that those typically aren't quantitated and we're not enriching for them in the purification process. There are, again, that's typically how most seasonal vaccines are produced. There are some different flavors of vaccines. So for individuals that are over 65, there are high dose influenza virus vaccines, which just means we have more HA contained in each of those vaccines. In some cases, there are vaccines that are adjuvanted, which means that we have added components that help boost the immune response. Again, typically, healthy individuals will not get those. Those are for older individuals that we know do not respond as well to influenza virus vaccines, or in some individuals that may have some sort of compromised immune system.
And just about embryonic cultivation in terms of influenza, because I know you said there's also cellular cultivation. Mm-hmm. Is the embryonic cultivation method preferable simply for the yield, or are there other reasons why embryonic cultivation is preferred over other methods?
It is primarily the yield, right? When you grow influenza virus in cell culture, it just doesn't grow to high titers. I'll point out, however, that there are, again, some problems with growing influenza virus in eggs. And one of them is I mentioned before, egg adaptations, right? Which is where the virus can adapt and change its proteins to be better suited to the egg environment, right? So that means that it makes changes in its surface proteins that are better suited to the environment of the embryonated egg. Sometimes when that happens, and then you take that virus and put it in humans, it's not as immunogenic, right? It doesn't elicit the immune response that we anticipate. And this is typically a problem for H3 viruses and can sometimes result, again, it can compromise the vaccine efficacy. So in some years where the vaccine efficacy isn't great against H3s, it's because of the egg adaptations that have happened in the process of producing the vaccine. So there are cell culture methods that are used to produce vaccines. Again, these avoid egg adaptations. You don't get as high a yield typically, but these are also, again, good for individuals who have egg allergies, allergies to egg proteins, that can't get the influenza virus vaccine that's typically produced in embryonated chicken eggs.
So how can our listeners today who are maybe interested in getting into virus research, how can they pursue that and pursue learning more about viruses?
Yeah, it's a great question. I would say, but I'll first and foremost plug the viral research that's going on at McMaster. We have a number of individuals doing lots of different research on virology from all aspects, from some of the things we talked about today in viral immunology and vaccine development. Mac has a new, as you may have heard, a new inhaled vaccine for SARS-CoV-2 that's currently in phase 1 trials. But the virology work spans through viral immunology to using viruses to fight cancer, to understanding basic principles about how viruses enter the cell and how the innate immune system responds. And so many of those researchers are affiliated with the Institute for Infectious Disease Research, the IIDR McMaster. So you can check out that website to learn more about their research. I'll also plug a podcast I like, which is called This Week in Virology. I don't know if you've heard of it. Some people call it TWIV for short, just the acronym of This Week in Virology, which again talks about all things viruses, talk about recent publications, they talk about current topics related to SARS-CoV-2 and the status of the pandemic. So I think those are both two good resources that individuals who are interested in viruses can explore.
So thank you for being with us today, Dr. Mullarkey. It was a delight to have you on.
Thanks for the kind invitation, Julia. It was a pleasure to speak with you.
Check out our Demystifying Research channel for more videos like this, and comment below any questions or thoughts that you had on the video.